In this chapter, I want to talk about the histology of accessory digestive organs. You know that we have some accessory digestive organs which can help better digestion. They are located around our digestive tube. As you see here, we have some salivary glands like parotid, submandibular and sublingual salivary glands. They are major salivary glands which we have around our mouth cavity, and they can make saliva. We have teeth and tongue as the accessory digestive structures, which can help making the food smaller and help mixing the food with our saliva. You can see liver in this picture. The liver has different functions, but it can make bile, and bile is stored in our gallbladder, and we can use it for the digestion of fats. In this picture, you can see right and left hepatic duct. They merge together and make common hepatic. Common hepatic and cystic duct from the gallbladder can make common bile duct. As you see here, the common bile duct come down and it merge with the main pancreatic ducts of the pancreas and make a short hepatopancreatic ampulla, and they open to our duodenum. The entrance of the hepatopancreatic duct to our duodenum is controlled by the hepatopancreatic sphincter or Oddi sphincter here. So all the times we don't have secretion of hepatopancreatic ampulla to our duodenum. But our pancreas has the other duct, which we call it accessory pancreatic duct. It opens to our duodenum alone before hepatopancreatic ampulla. This duct doesn't have a sphincter and it's open all the time and help the pancreatic juice to come to our duodenum. And in this part, I want to talk about the histology of some of these accessory digestive organs for you. The first one which I want to talk about is our tongue. In this picture, you can see mid-sagittal and longitudinal section of the tongue. Our tongue is covered by mucosa from outside. You can see the epithelium and lamina propria on the dorsal surface and ventral surface of the tongue. And the core of our tongue is made up of a group of skeletal muscles, which are located in different orientation. Some of them are longitudinally located, some of them are oblique, some of them are transverse. Because of that, we can move our tongue in different directions. The mucosa of the dorsal surface of our tongue can make some protrusions, some projections. These projections can make lingual papilla. We can see lingual papilla on the dorsal surface of our tongue and on the tip of the tongue. We don't have any papilla on the ventral surface. And you know that your ventral surface of tongue is completely smooth. We have different types of papilla. One of them is called filiform. It's thread-like. One of them is conical. It's cone-shaped. These two papilla are physical papilla or mechanical papilla. They can only help the bolus to go backward in our mouth cavity. The other papilla, like fungiform, foliate, and circumvallate papilla, contain taste buds, and we can receive the sense of taste with them. For now, you don't need to know the name of the papilla, but you need to know that some of them can only work mechanically, but some of them can do sensation for us. In the tongue, you can see minor salivary gland. This salivary gland is called lingual gland, and the lingual gland can secrete both serous and mucus, and we have some ducts which can convey the secretion to our mouth cavity. This is the higher magnification of the tongue. In this higher magnification, you can see the epithelium which covers the surface of our tongue. This epithelium is a stratified squamous epithelium, and you can see lamina propria or connective tissue beneath it. This papilla is the sensory papilla. It can do sense of taste for us. And we have some pale structures in the wall of this papilla. These structures are called taste bud. In the taste bud, we have some sensory cells or gustatory cells which can get sense of taste, and some supporting or sustentacular cell which can support the sensory cells. Then they are connected to the nerve fibers, and the nerve fibers can bring the information to our insula, the lobe of our brain, and then we can get the sense of taste. In this picture, you can see the glands here. Here, you can see the larger magnification of the taste buds. When you look at them, they are paler than the epithelium, and their shape is very similar to the onion. And you can see different types of cells inside. I don't want you to know the name of the cells, but the clear one are gustatory, and the darker one are supporting or sustentacular cells. On the surface of this taste bud, we have a pore which we call it taste pore, and some microvilli or taste hair come out of the taste pore to receive the sense of taste. Here you can see the microscopic view of the tongue, and you have 1, 2, 3, and 4 papilla here. All of them are sensory papilla. They are projection of our tongue into our mouth cavity. You see that they contain pale structures on their wall. These pale structures are called taste bud. The epithelium which cover these lingual papilla are stratified squamous epithelium. Associated with this papilla, you can see some glands here. They are lingual salivary glands, and these lingual salivary glands contain the excretory ducts, which can help the sending the secretion into our mouth cavity. And beneath them, you can see skeletal muscles in different orientations. And this connective tissue, which is located between epithelium and the muscle, is the connective tissue of lamina propria. Here we have areolar to dense irregular connective tissue. In this picture, this is the other lingual papilla. You can see the taste buds here, the pale structures in the lateral wall, and the epithelium is a stratified squamous epithelium. Here you can see the lamina propria, and the muscles which you can see here are skeletal muscles. This is the other picture of lingual papilla. You have 1, 2, and 3 lingual papilla. On their lateral wall, you can see taste buds, the pale structures, onion-shaped pale structure. The epithelium of the lingual papilla are stratified squamous epithelium, and beneath this stratified squamous, you can see the lamina propria. Here we zoomed in the lateral surface of the papilla, and you can see 1, 2, 3, 4, 5, and a part of 6 papillas, taste buds here. As I told you, they are onion-shaped and they contain taste pore. The next structure is the microscopic structure of salivary gland. In the lecture, I will talk about salivary gland completely for you. The parotid salivary gland is composed of only serous secretion. The submandibular and sublingual salivary glands have mixed secretion. It means that they have both serous and mucous secretion, but the ratio of serous and mucous in these two glands are different from each other. In this picture, you can see serous and mucous acini. Acinus and the plural of the acinus, which is acini, are the group of cells which come together and make cluster. Here is a cluster of cells. They come near each other and they can make one group. If the nuclei of the cell is located in the base of the cell and the nuclei is flat, this acinus is called mucous acinus. And if a group of cells which we have near each other contain round nucleus in the center, like the one which I draw here, we call it serous acinus. In the picture, which you can see here, we have mucus, because you see that the nuclei are flat on the base, and the serous acinus. This group of cells send their secretion to a very small, tiny duct. These tiny ducts are called intercalated duct. Intercalated duct can make larger duct, which we call it striated duct. We call it striated because they have striations or lines. If I magnify the picture for you, you see the lines in the base of the cells. Because of that, we call this duct a striated duct. Intercalated and striated duct have simple epithelium. Their epithelium has only one layer. Then the striated ducts of the different parts combine together and make one large duct. We call it excretory duct. And the epithelium of the excretory duct is stratified. You see that we have more than one layer here. And this duct can convey the secretion into our mouth cavity. Around the serous and mucous acini, we have a group of cells which we call them myoepithelial cells. The myoepithelial cells are associated with the epithelium of the acini. Between the epithelium and the basement membrane, they have the characteristic of muscles. It means that they have actin and myosin. And contraction of these muscles can squeeze the acini and help to send the secretion to intercalated duct. So this is the general structure which you can see for the salivary gland. Here is the microscopic view of parotid salivary gland. All of these groups are called acini, and they are serous acini because they have a round nucleus. They send their secretions to a tiny duct, which we call it intercalated. Intercalated can make larger duct, which we call it a striated duct. We need to zoom in these ducts more to see the lines or striations. can see the microscopic view of parotid gland. The parotid gland, which is located anterior and inferior to our ear, is the serous gland. This gland is composed of thousands of the serous acini. They can secrete saliva, and the saliva which is secreted come to intercalated duct. We can see intercalated duct by this magnification. Intercalated ducts can make larger duct like this, which is a striated duct. And a striated duct can make very large duct, which we call it excretory duct. And excretory duct can send the secretion into our mouth cavity. The excretory ducts are located in the connective tissue. This is the other microscopic view of parotid gland. In this image, you can see different serous acini. They send their secretion into intercalated duct, and intercalated ducts can make a striated duct. This is one, and this is two striated ducts, which you can see in this picture. The next structure which I want to talk about is the pancreas. Here you can see the pancreas. You need to know that the pancreas is a double organ. It means that it has two types of secretion. One type of secretion is endocrine. The secretion goes to our bloodstream, and this group of cells and this one, which contains blood capillaries near themselves, are endocrine. The other one is called exocrine. The acinus contain duct, and the duct can send the secretion to our small intestine. So our pancreas has two parts. Endocrine part, which is about 1% of our pancreas, we call it pancreatic islets or Langerhans islets. And here you can see exocrine part, which we call them pancreatic acini. In the pancreatic acini and exocrine part, like the salivary gland, we have intercalated ducts, then we have a striated duct, and then we have large excretory duct. Here is the microscopic view of the pancreas. You can see two colors in this picture. One color is pale, one part. We call them pancreatic islets. One part is deeper in the color, which are the pancreatic acini. The pancreatic acini, all of them can make serous secretion. They make pancreatic juice, and the pancreatic juice can come to our duodenum. But the Langerhans islets can make insulin and glucagon, and these two hormones can regulate the level of sugar in our bloodstream. This is the histology of pancreas. The pale part is islets of Langerhans or pancreatic islets, and the deeper parts are pancreatic acini. Here is the other picture of pancreas. Here you can see the pancreatic islets or Langerhans islets, about 1% of our pancreas, and these deeper in color parts are called pancreatic acini. Each of these group is one acinus. They can make pancreatic juice. Pancreatic juice contains protease, lipase, amylase, and nuclease, which can digest different groups of nutrients for us. Here is the other picture of the pancreas. In this picture, you can only see the pancreatic acini, and between these acini, you can see a pancreatic duct which can convey the secretion into the duodenum. The secretion of our pancreas is alkaline in its nature. It comes to the duodenum. The chyme which come from our stomach is acidic, and this alkaline nature of pancreatic juice can neutralize the acidity of chyme for us. And here you can see the other picture of the pancreas. You can see these pancreatic acini. Each of them is one acinus. And here you can see one pancreatic duct between the acini. The other picture of pancreas. In this picture, you can see two pale parts. They are pancreatic islets, and the deep in color parts are pancreatic acini, which can make pancreatic juice for us. And the last structure which I want to talk about is the liver. You know that we have 4 lobes for our liver: right, left, caudate, and quadrate. Each of our liver lobe is composed of smaller parts, which we call them lobule. Here you can see one lobule. The liver lobule are hexagon in their structure, as you see in this picture. Each of these hexagons contain one vein in the center, which we call it central vein. And on the corner of this hexagon, you can see the structure which we call it portal triad or portal area. In each portal area, you have 3 structures. Because of that, we call it triad. One of these structures is a branch of hepatic artery, like this one or this one. The other structure is a branch of hepatic portal vein, like these 2. And the last structure is called bile duct. And this is bile duct. So in each corner of the hexagon, we have portal triad or portal area. Each portal area has a branch of hepatic artery, a branch of hepatic portal vein, and a branch of bile duct. Around the central vein, you can see the cells in parallel organization. All the cells which you can see here are hepatocyte or liver cell. And between the hepatocyte, you can see blood sinusoids, or you can see tiny bile ducts, which we call them bile canaliculi. So between the cells, we have sinusoids or hepatic sinusoids and bile canaliculi. Here is the microscopic structure of liver lobule. You can see these hexagons, and you can see the central vein in the center. In the corner, you can see portal triad or portal area. Each portal triad has one bile duct, one branch of hepatic artery, and one branch of hepatic portal vein. In the other one, you can see the hepatic artery, bile duct, and hepatic portal vein. In the other one, hepatic portal vein, bile ducts, and branch of hepatic artery. The cells around the central vein are hepatocyte. Between the hepatocyte, the clear area are blood sinusoids or hepatic sinusoids. This is the other picture of the liver lobule. In this picture, again, you have 1, 2, 3, 4, and 5 lobule. You have central vein in the center, and in the corners, you can see portal triad or portal area. This is the histology of the liver. You can see different hepatic lobule here. They are hexagon structure. As I told you, they have central vein in the center and the parallel organization of the hepatocyte around. And in the corner of this hexagon, you can see portal triad. This is the other picture of the hepatic lobule. You can see many hepatic lobule here. This structure in the center is the central vein. You can see the portal area or portal triad on the corner. And these are the hepatocyte. Between the hepatocyte, we have hepatic sinusoids. This is the higher magnification of the central vein, and these are the hepatocyte. The interesting thing about hepatocyte is most of them, not all of them, has 2 nuclei, like this one, this, or this. But we have some cells which only contain 1 nuclei. Between these cells, all of these clear area represent the hepatic sinusoids. In the lecture, I will talk about the flow of blood and bile in the liver completely.